In this video, we're going to explain DNA replication. So we're going to go through the whole process, exactly how it works, the enzymes that are involved, and explain what semi-conservative replication means. So let's start by thinking about which stage of the cell cycle does DNA replication actually take place in. So you'll remember from GCSE that there are 3 stages of the cell cycle. So stage 1, which takes up about 90% of the time on average of the cell cycle, so it's the longest phase. This is interphase, and this is when DNA replication occurs. It's actually during the synthesis phase of interphase. So you might learn that interphase has 3 phases. It has G1, which is the growth 1. It has the synthesis phase, and then it has the G2, which is the growth 2 phase. But DNA replication specifically happens during the synthesis phase of interphase. Now, if you're doing AQA biology, you don't really need to know that extra detail. So we can just say that DNA replication occurs during interphase. The other stages of the cell cycle, stage 2 is mitosis or nuclear division, where the chromosomes are pulled to opposite poles and separated to form 2 new nuclei. And then we have stage 3, which is cytokinesis. This is where the cell actually divides. So the cytoplasm divides and the 2 new cell membranes form to form 2 genetically identical daughter cells. We're going to focus obviously on stage 1 because stage 1 is where DNA replication occurs, which is what this video is all about. Let's have a look at the process. So we've got a full overview here. You can see the chromosome. We've kind of zoomed in on the DNA that forms that chromosome. Remember, in eukaryotic cells, the chromosome is formed from DNA, which is wrapped around histone proteins. You can see the replication fork. You can see where the DNA strands have been separated, and you can see the new DNA being synthesized. Can we identify the 2 enzymes used in DNA replication? Have a little look at the diagram. The first one is here, so we're going to talk about DNA polymerase. The second one is here, which is DNA helicase. Now, there are some other enzymes involved, like DNA ligase, but for A-level biology, we just focus on these 2 enzymes. So when you're answering questions about DNA replication, make sure you name the enzymes, DNA helicase and DNA polymerase, and make sure you describe their job in detail using specific scientific language. So let's go ahead and do that together. We're going to go through it step by step now, focusing on the enzymes, obviously. So the first stage, we're going to talk about DNA helicase. So as you can see on the diagram, DNA helicase is breaking the hydrogen bonds, but we can say a little bit more than that. So we can say that DNA helicase breaks the hydrogen bonds, but we're going to add on between the complementary bases. So we're telling the examiner where those hydrogen bonds are. So it's moving along the DNA, it's breaking those hydrogen bonds, which do break quite easily because they are weak into molecular forces. Breaking those hydrogen bonds, which are between the complementary bases, and this separates the 2 strands, or unwinds the DNA to separate the 2 strands. Moving on to the second diagram, both strands act as templates. Now, this is different to transcription, where only 1 strand acts as a template. But remember, that's when we're making mRNA. So we'd also be using RNA polymerase in that process. So we're going to be using DNA polymerase here. We're making DNA, and both strands act as templates in DNA replication. We can see on the diagram that 3 nucleotides are moving in and lining up with the exposed bases on those 2 template strands. So let's write that next. So 3 nucleotides line up with the exposed bases on the template strands. And we can also add, because it might be worth a mark, according to the base pair rule. So they don't just line up in any old order. Obviously, the order of the bases on the two template strands is going to determine the order that these new nucleotides line up in. And we can even give an example. We'll give all the examples of that base pair rule. So if there's an A on the template strand, obviously that would pair with a nucleotide with a T. If there's a T on the template strand, it would pair with a nucleotide with an A. If there's a C on the template strand, it's going to pair with a G. And if there's a G on the template strand, it's going to pair with a C. So we're giving examples there of the base pair rule and how these free nucleotides are going to line up with the template strand. Okay. Once they line up, obviously hydrogen bonds will reform between those bases, but that does not require an enzyme. Hydrogen bonds are kind of like attractive forces, and it's just because the bases are complementary that those hydrogen bonds will form, so that does not actually require an enzyme. Let's move on to look at the final couple of stages then. This is where we're going to mention DNA polymerase, okay? Another important enzyme. So once the free nucleotides have moved in, lined up with the exposed bases according to the base pair rule, DNA polymerase is going to join the nucleotides together. But again, we can add on a little bit more than this. So DNA polymerase joins, we quite often say the adjacent nucleotides together. You don't have to in an exam. You can just say DNA polymerase joins the nucleotides together. But what number are we up to? We're up to number 6. Trying to number it so you can get yourself a nice little set of notes. DNA polymerase joins the nucleotides together with, let's name the bond. We love naming a bond in biology. So with phosphodiester bonds, and I'm also going to say that are formed via condensation reactions. Now, phosphodiester bonds are strong covalent bonds. They're formed via condensation reactions, and the enzyme DNA polymerase is catalyzing these reactions. Obviously, it will involve the removal of water every time we make one of these phosphodiester bonds. But what I want to really stress here is DNA polymerase is not joining the bases together. It's not joining the strands together because the hydrogen bonds between the strands don't require an enzyme to form. So what DNA polymerase is actually doing is joining one of these nucleotides to the next on the new strand, okay? And it will be doing the same on this strand, but just moving in the opposite direction because the strands are anti-parallel. So it's making phosphodiester bonds between these nucleotides on the new strand. So it's basically forming the sugar phosphate backbones, okay, via condensation reactions. So the process is now pretty much complete. That's the amount of detail that you need for A-level biology. So don't worry about learning anything extra that your teacher may have taught you. If you're just at A-level level. Let's just finish it off though, because we can sometimes get credit for saying DNA replication is semi-conservative, which we can kind of see on this last diagram here. What that means is each DNA molecule that we've produced will contain one original or old strand and one newly synthesized or one new strand. Because you can see the original strands here in black, and they were the strands that both act as templates. And then you can see in the middle in purple, the new strands that have been joined together with those phosphodiester bonds using DNA polymerase. So we say it's semi-conserved because it's kind of half original, half conserved, or half kept the same, and half of the molecule consists of that new strand. There'll be genetically identical copies. ready for the cell to go into the next stage of the cell cycle where we have mitosis. And obviously this will result in genetically identical cells because they both have exactly the same DNA. I hope you found that video useful, and hopefully we're going to get a nice 5 marker on just describing the process of DNA replication, because now we've been through it in this detail, that would be super easy to get all the marks. Just remember, name the enzymes, say what they do using really clear language, talk about the bonds that they break or they make, and you'll be absolutely fine.